So this work is in collaboration with Astrid, and it's really a testament to her dedication to science and to her grad students that in the very 11th hour of her career as a scientist, she went out with me last year, and we spent 5 weeks working every day in the field while Jim was reporting in that the fires of Arizona were bearing down on the cabin and portal. And so, yeah, and this is, I'm going to talk about our work on that today. So I want to talk to you about how examining female mate choice can actually provide insight into what selective pressures lead to sympatric speciation. And specifically, I'll be discussing how our examination of female mate choice in a sympatric species flock of pupfish has indicated that predation and some frequency dependent effects reinforces premating isolating mechanisms. So, sympatric speciation is the divergence of one species into two or more, what's going without me, sorry, into two or more lineages without geographic isolation. And there's agreement that ecological processes such as trophic differentiation to exploit different diets and niches are the driving forces of sympatric speciation. However, the premating isolating mechanisms that result in assortative mating and thus genetic divergence, right, are still poorly understood. So the role of sexual selection in sympatric speciation is still debated, since assortative mating by itself does not necessarily result in speciation. However, sexual selection may facilitate differentiation if assortative mating is based on traits reflecting ecological relationships, such as diet or size or habitat. For example, the color morphs in Midas cichlids. So Gavrilets had suggested that so-called magic traits, which function both in ecological differentiation and mate choice, can lead to rapid speciation. If females identify conspecific males based on those traits associated with ecological characteristics such as diet, etc., then a genetic association between the ecological and the mating traits could lead to rapid speciation. And studies of sympatric species flocks have inferred the existence of assortative mating. But there are relatively few studies that have actually examined the premating isolating mechanisms. And such studies are really important because they provide insights into the underlying mechanism of disruptive selection, right, that leads to speciation. So the role of predation in sympatric speciation isn't quite so straightforward. We know that predators can inhibit the evolution of premating isolation because of the increased survival costs associated with searching for mates in heavily predated environments, and results in relaxed sexual selection. However, predators have been shown to enhance the formation of premating isolating mechanisms in some systems. So Nosal and Krebsey have recently shown that adaptive divergence in the color patterns linked to host plant usage of two species of walking stick insects was greater in the presence of predators. And similarly, limnetic and benthic sticklebacks show greater divergence in the presence of predatory fish. So in this kind of situation, predator-prey interactions could actually accelerate the evolution of pre-mating isolation among sympatric species if the presence of a predator reinforces the underlying mechanisms that promote assortative mating in the prey species. Frequency-dependent selection can also facilitate assortative mating in sympatric species. With high frequencies of conspecifics, probabilities of encountering individuals of another species are low, and thus selection for cues promoting assortative mating is relaxed. However, if the frequencies of conspecifics are low, then encounter rates with heterospecifics are high, which results in more selective pressures on assortative mating. So this leads to the expectation that pre-mating isolating mechanisms will be stronger in species that are found at comparatively lower frequencies. So to examine the role of predation and frequency dependence on sympatric speciation, we examined cues used in species recognition and assortative mating in a sympatric species flock of pupfish from San Salvador Island in the Bahamas. And we focused on these two, as of yet undescribed, sympatric species, and they're comprised of morphologically and genetically distinct generalist Detritivore on the top, and a predatory scale eater which predates upon the Detritivore on the bottom there. So we collected the fish from the not good part of the Bahamas, the interior. I know everybody's like, oh, you went to the Bahamas. Yeah, no. We saw pretty parts of the Bahamas, but occasionally. From Ospre Lake and Little Lake. And this last year we went back 2 weeks ago. We just got back and went and repeated the efforts at Crescent Lake, but I won't talk about that today. And these two species, the microsatellite, nuclear, and topographical data all indicate that there has been some gene flow between Little Lake and Ospre Lake, but it's restricted. So to determine what mechanism maintains pre-mating isolation, we examined the cues used in female mate choice. And while these species are morphologically distinct, so we definitely wanted to examine visual cues. There's also the only other sympatrically speciated flock of pupfish uses chemical cues in their mate choice. So we additionally examined olfactory cues used in female mate choice. And then finally we wanted to look at the effects of predation on mating preferences in prey, in the Detritivore species, and also the effects of frequency dependence on mating preferences in both species. So we hypothesized that the abundance of the predator species, the scale eater, determines the strength of pre-mating isolating mechanisms in the Detritivore. And we predicted that those pre-mating isolating mechanisms will be better developed in populations of Detritivores that experience the higher levels of predation by scale eaters than those where the scale eaters are less abundant. And additionally, we hypothesized that pre-mating isolating mechanisms are frequency dependent and will be better developed in the less abundant predator species than in the more abundant generalist prey species. So 5 weeks, Astrid and I spent together every day, and I'm happy to say that I came back still liking my advisor, actually liking my advisor more, I think, than when we went out there. So we collected fish by minnow trapping and seining and suckering other pupfish researchers into helping us seine. And we also, when we failed to find fish on our side of the lake, we got a canoe from the field station that we were staying at, which you can see was in very nice condition. And the two of us rowed it across the lake, which took about an hour, I would say, for us to row. And Astrid was up in the bow, and she was rowing so fast that I couldn't keep up with her, and I would have to tell her, Astrid, slow down, and I would try so hard. And finally, I got really tired of my advisor putting me to shame in my rowing abilities, so I asked her one day if she would let me rig our seine into a sail, and she looked at me and said, okay, because Astrid always lets me try things, and whether or not she thinks they'll work. And so I rigged a sail out of the seine, and we sailed back, and Astrid spent the whole time with her face in the lake staring at the fish. So it was good for both of us. So we brought the fish back to the research center where we did our behavioral trials at the site. We placed 2 males of each species into 2 separate clear plastic containers inside of a large round kiddie pool. And then we placed a female of one of the species into the round pool, and we measured the amount of time that she spent associating with males of either species. And then we repeated this for the females of the other species, and then we collected the water from the males, which had been sitting overnight for 24 hours before the visual choice trials. We collected the water from the males for use in the olfactory trials. So to determine if the females used olfactory cues to detect conspecifics, each female was placed into a clear aquarium, and then water, control water and conspecific water was dripped into each side of the tank. And the time that she spent in either side of the tank was recorded. She was then moved to another tank, and heterospecific and conspecific water was dripped in, and time was recorded. So we found that there was about 1 scaliter per 500 detritivores in Little Lake, and 5 scaliters per 500 detritivores in Ospre Lake. And we thought about Jim and his fondness for counting animals as we were doing this. We found that detritivore females from Ospre Lake spent significantly more time with conspecific males, and those from Little Lake did not visually discriminate between conspecific and heterospecific males. Female scalators prefer the site of scalator males to detritivore males, and additionally, scalator females from Little Lake spent significantly more time with conspecific males than those from Ospre Lake. Detritivore females from Ospre Lake but not Little Lake spent more time with the odor of conspecific males, but did not discriminate between the odor of conspecific males in water. And scalator females did not discriminate between the odors of conspecific males, heterospecific males, or water. However, between lakes comparison shows that females from Ospre Lake spent significantly more time with odor presentations in all treatments than did females from Little Lake. So, the species isolating mechanisms in these species are based on visual cues, which play a major role in female recognition of conspecifics. The 2 species differ in shape, with particularly the males. Detritivore males exhibit this large hump-like nape, whereas both sexes of the scalators are very fusiform in shape. They're like little scale-eating torpedoes. They also differ in jaw morphology, and then the male breeding coloration, with the scale-eater males being almost jet black, and the Detritivore males can show a range with being jet black all the way to a more stereotypical pupfish bright blue nape with red or orange underbelly. The development of pre-mating isolating mechanisms is frequency dependent. Selection for positive assortative mating is stronger in the less abundant scale eater than in the more abundant Detritivores in both lakes. Additionally, the strength of female visual preference for conspecific males was stronger in Little Lake, where scale eaters were less abundant than in Ospre Lake. And the responses of Detritivore females from 2 lakes to conspecific and scale eater males suggest that the abundance of predators also affects the development of pre-mating isolating mechanisms in the prey species. Only females from the lake with a higher frequency of scale eaters showed a visual response to conspecific males. And finally, this really indicates that experimental studies of underlying mechanisms of species recognition, which leads to assortative mating in sympatric species, really provides unique insights into the underlying mechanism of sympatric speciation. And so just showing that sympatric species have genetically diverged, and from that saying, well, there's probably some pre-mating isolating mechanisms going on, really doesn't give you any insight into what the underlying ecological factors that likely led to that sympatric speciation event are. Okay, so thank you guys for your time.